My name is Dr. Paul Richardson, and it's my pleasure to give you a quick summary of our presentation at ASCO on the DREAMM-5 platform trial, which is a very novel and exciting multi-center international effort looking at combining the first-in-class antibody-drug conjugate, so-called Belantamab mafodotin, in combination with a number of other fascinating novel agents for the treatment of relapsed and refractory multiple myeloma. Now, by way of background, Belantamab, as it's called, or Belantamab mafodotin, its other big name is GSK 2857916, but in the interests of brevity, I'll stick with Belantamab, if I may. This is a first-in-class B-cell maturation antigen-binding humanized monoclonal immunoconjugate. And what that means is that it's a so-called antibody-drug conjugate that delivers to BCMA-expressing cells a potent cytotoxic. And this actually is very interesting because what it does is, by doing this, it has multimodal activity, and it activates the immune system against the disease, but also delivers this specific cytotoxic and triggers so-called immunogenic cell death. Now, the mechanism of action of Belantamab are quite complex, but suffice to say that there is this effect on what we call ADC, and then there's this effect on what's called ADCC and ADCP, and this leads to immunogenic cell death, not least of which because there's actual delivery of this cytotoxic component that further enhances the anti-myeloma effect of the platform. Now, with the success of Belantamab seen as a single agent, what DREAMM-5 is seeking to do is to combine Belantamab with a number of novel approaches to further enhance its activity. And how we set about doing this is combining it with three other agents. And the first is a sub-study, as we call it, combining Belantamab with so-called OX40 antibody inhibition. So this is with a drug that goes by the name of GSK 998, which is a humanized wild-type IgG1 anti-OX40 agonistic monoclonal antibody that binds to this costimulatory OX40 receptor, and this is expressed primarily on activated CD4 and CD8 positive cells. Now, because of that, this can then further enhance the activity of Belantamab. So the intention of sub-study 1 is to combine GSK 998 with Belantamab and see if this is first and foremost well tolerated, and second of all, does this have a good effect or an enhanced effect against the myeloma. Now, it's important to note that we've studied this preclinically, in which in a mouse model, we've combined Belantamab with an OX40 surrogate antibody, and the activity seen has been remarkable. So there's a very hopeful preclinical experiment to justify this clinical approach to help patients. Now, sub-study 2 is combining Belantamab with a different monoclonal antibody. This one is called GSK 609, and it's a humanized anti-ICOS IG4 monoclonal antibody selected for its nanomolar binding, which means low level, but its potent agonistic activity on ICOS-expressing CD4 and CD8 effector T cells. Now, these really matter because when you combine them with Belantamab, they are particularly potent against myeloma preclinically. So we're very hopeful that that combination will work well too. And then finally, in sub-study 3, we're combining Belantamab with what I think is a particularly interesting approach. Gamma-secretase is an enzyme that cleaves membrane-bound BCMA from tumor cells, and by so doing can interfere and limit the efficacy of BCMA-targeted therapy. Now, nirogacestat, as it's called, or NIRO for short, is, and that's NIROGACESTAT is how we actually fully spell it. NIRO is a novel gamma-secretase inhibitor that inhibits NOT-related T and B cell maturation in vitro. It's already bioavailable, and so by inhibiting gamma-secretase activity, it can increase cell surface levels and availability of BCMA, and the preclinical evidence for this shows that it's really quite potent. So the third sub-study is combining NIRO with Belantamab to see if that further enhances activity. Now, all three sub-studies are currently open to accrual, and in this study, we are also offering patients Belantamab alone as a part of a control to demonstrate convincingly that we're actually making a difference for patients with these additional approaches, not least of which because belamaf itself obviously has substantial activity, and we're hoping very much it will gain accelerated approval sometime later this year. But by combining these novel approaches, we hope to then be able to take forward combinatorial strategies that then can further help patients in the future. As people I think are aware, belamaf is generally well tolerated. One of the bigger challenges is the ocular toxicity, which is generally very manageable actually with dose and schedule adjustment. And in this particular trial, we're taking forward lower doses of belamaf in a deliberate effort to seek to minimize any ocular toxicity that may occur, and yet retain efficacy, and in fact hopefully have enhanced efficacy by virtue of combining it with these novel drugs, specifically the monoclonal antibody targeting OX40, the other monoclonal antibody targeting ICOS, and last but no means least, the potential benefit of gamma-secretase inhibition. So just want to conclude by especially acknowledging my co-investigators and our super team of physician scientists who are part of the GSK team and all the other study staff involved. And as I mentioned, the study is ongoing with enrollment now underway. Thank you very much for your attention.